Hi everyone. Good to see you guys have joined. I'll start the class now. It's mostly going to be looking at the last topic about adrenal glands, and also now looking back and revising some key and core concepts that were covered in this course. I hope everyone can hear me. Um, yeah, so I'll get started, and please feel free to ask questions in between if you have anything you're confused about, or you can send questions in the chat box, and we'll also discuss some questions so you can send the answers in the chat box as we go along. Yeah, great. Um, Ideally, it would be better if others could mute themselves so that um, it doesn't disturb the class. And then if you need to ask a question, you can unmute and ask me then, right? Okay, so I'll just share my screen. Yeah, I hope you can all see what I'm sharing. Great. So, yeah, welcome everyone to week 11 of the problem solving sessions for human physiology. And today we will continue from where we left off in the last class, where we were talking about adrenal glands and the steroid hormones that are secreted by the adrenal glands. So we covered some important concepts about the structure, anatomy, and morphology of this gland, right? So it has these different zones or layers in its organization, and these zones have specific functions. They secrete specific hormones that have very important functions in the body. And we talked about them. So, right, the outermost layer that you will see in the adrenal gland is this connective tissue capsule that covers it and protects it. And then below that, you have the outermost zone, which of secretion, essentially, of synthesis and secretion of steroid hormones, which is the zona glomerulosa. Inner to that is the zona fasciculata, and then further inwards is the zona reticularis. And these three make up the cortex. The adrenal cortex is made up of these three zonas. And then innermost is the adrenal medulla, which also has its important functions, as we discussed over here. So this is just about the adrenal cortex, where you have the zona glomerulosa, the outermost layer, right? It's very important because it is the site of production of aldosterone, which is a mineralocorticoid and a very, very important enzyme and part of the RAS system. And it is important for regulation of blood pressure. So without it, actually, it could be very lethal. So next part, the zone is the zona fasciculata. So that is sort of sandwiched in between these two zones, glomerulus and reticularis. And these cells in the fasciculata are responsible for producing glucocorticoids. So above it, you have mineralocorticoids. Now you have glucocorticoids. And one very well-known example is cortisol, right? The stress hormone. So this zona fasciculata is the largest of the three layers, and it's about almost 80% of the volume of the cortex is the zona fasciculata. And it synthesizes and secretes this very important hormone called cortisol. Okay. Then inner to that, you have the zona reticularis. This is the innermost layer of the adrenal cortex, and it lies adjacent to the medulla, and it produces these hormones called androgens, which you must have heard of before. Uh, Sorry, does someone have a question? Okay, if you have a question, please um, just unmute and say so, or send a message and I'll see it eventually. Cool. Right. So Yeah, so this zona reticularis produces androgens. Some examples are DHEA and DHEA sulfate, right? So these are commonly um, uh, acid enzymes, sorry, hormones. Um, just a second. Yeah. And yeah, so this has a more sort of irregular appearance with respect to cords and clusters of cells. And it's sort of brownish because of this pigment called lipofuscin. So then we also talked about steroidogenesis last time, where we talked about how these hormones are synthesized. Steroid hormones are synthesized within the adrenal gland. So essentially, the main sort of starting material for the steroid hormones is cholesterol. So it's this 27 carbon molecule, and the first common step before it sort of divides up into the different regions of the adrenal cortex based on what needs to be synthesized. The first common step is converting cholesterol with the help of cholesterol desmolase, which is also called CYP11A1 or P450 SEC. It's all of these are synonymous. So this enzyme is super important for converting cholesterol into pregnenolone, which is a 21 carbon compound, and that happens because there's a 6 carbon side chain that's removed with the help of this enzyme, and this occurs in the mitochondria. And once you have pregnenolone, then it enters into these different pathways where it eventually, with the help of these specific enzymes like CYP17 in these two, fasciculata and reticularis, and CYP21A2 in the zona glomerulosa, it eventually leads to either aldosterone biogenesis in the zona glomerulosa, which is your mineralocorticoid, or glucocorticoids such as cortisol in the zona fasciculata, or through a series of steps into DHT and estrone. This happens actually. There's a portion of the androgen pathway that happens actually in the peripheral tissues. So DHEA and androstenedione, these are actually weak androgens, and when they reach into the peripheral tissues, they further undergo some steps where they become more potent androgens, um, such as DHT via 5-alpha reductase, or estrone, which are the estrogens basically, via aromatase. So these enzymes are there in the peripheral tissues, not in the adrenal gland, basically in other tissues where they undergo these changes. So this is how you start from the same material, but then it sort of divides up zonally in the adrenal cortex, and you get these different end products. So the main function for mineralocorticoids, as we talked about, is the blood pressure and the balance of sodium and sodium retention, water retention, potassium secretion, etc. And very important function for cortisol is the increase of gluconeogenesis, making glucose from basically like non-carbohydrate sources, so like using amino acids, for instance, for making glucose, or fats. It also is important for protein and fat mobilization, sometimes effectively for gluconeogenesis, immune suppression, stress response, which we very well know about. We've heard about it, I'm sure, a lot. When people talk about stress and stress management, they talk about cortisol levels quite a lot. And tests in animals like rats uses also like assays and ELISAs to look at the level of cortisol, but in rat models, it's known as corticosterone. And finally, you have the androgens in the reticularis, and these are important for secondary sexual characteristics in humans. Great. So we also, as I mentioned, talked about cortisol, which has many functions, very pleiotropic, some of which are increasing blood sugar because you want to make more glucose available for use. heightened memory and attention, increase in blood pressure, which is why people may relate like stress and blood pressure because cortisol may have a role to play there. Decreased sensitivity to pain, immune suppression, and decreased serotonin. So that's maybe another reason to link sort of mood disorders with cortisol levels. Right. So, yeah, so we'd left off at a certain number of questions. So I will now show you the question if for anyone who's not joined before, and we will discuss the answer. So you guys can send the answer um, in the chat, um, and we can discuss it. Right. So the first question here is the primary hormone that regulates cortisol secretion is which of these options? So do you guys want to try it out? You can try and we can discuss. Anyone want to try? So you just have to send the option that you think it is A, B, C, or D. Um, if you're like not able to text, you can also like unmute and say the answer if you feel that more comfortable. Yeah, I'm getting some answers here. Does anyone else want to try? Yeah. So great job. The correct answer is option B. This is what all of you guys have said. So great job. ACTH is the correct answer. So ACTH, as you guys know, is adrenocorticotropic hormone. Okay, which and it's one of the primary regulators of cortisol secretion. So where does ACTH come from, right? It's released from the anterior pituitary in the brain, right? And it acts on the zona fasciculata region. So ACTH essentially stimulates steroidogenesis because it can cause the upregulation of enzymes that are involved in cortisol synthesis. And ACTH in turn is also, its secretion is also controlled by the CRH from hypothalamus. So that's essentially the hypothalamic pituitary adrenal axis or the HPA axis, where you have hormones from hypothalamus controlling the pituitary, anterior pituitary, such as in the release of ACTH, which then controls a specific endocrine organ like the adrenal gland via ACTH. So that circuit is called the HPA axis, which we discussed in the last class, I believe. Great. All right, so let's do another question. So ACTH is secreted from primarily where? So I think this is, I think I gave away this one, but let me know if you guys remember. Does anyone else want to try answering? Yeah, yeah, great job. So I'm seeing that people have mostly got it right. It's option C. which is the anterior pituitary. So it's produced by these cells called corticotroph cells, which are there in the anterior part of the pituitary gland or the anterior pituitary. So this it's these cells, like, they are stimulated by the hypothalamus via the CRH that I mentioned, right, which is corticotropin releasing hormone, or CRH. So the hypothalamus secretes CRH, corticotropin releasing hormone, which acts on these cells, the corticotroph cells in the anterior pituitary gland, which then releases ACTH. And then ACTH will travel via the circulation, hit the adrenal cortex, and stimulate cortisol synthesis and release over here. So this is very important for proper endocrine regulation of the adrenal glands. Okay, let's do another question. Cortisol exerts its effects by binding to which of these receptors? What do you guys think? Anyone wants to try? Right. Great. So I've got the correct answer here. Good job. It is indeed option C, which is nuclear receptors after cytoplasmic binding. So what does that mean? Essentially, there is an intracellular receptor mechanism involved here, right? So cortisol itself is quite lipid soluble, right? Because we learned about how these steroid hormones are quite lipid soluble. And so therefore, cortisol actually diffuses through the plasma membrane. And when it does that, it goes inside the cell and it binds to these glucocorticoid receptors that are just present in the cytoplasm. So they're GRs, right? They're present in the cytoplasm. And then this complex of the hormone bound to its respective receptor. This translocates to the nucleus. So this bound complex, hormone receptor complex, translocates to the nucleus. And there this then binds DNA at certain response element regions that regulate gene transcription. So this is how sort of there's a direct effect, like it's almost a genomic mechanism, right? So it's a slower mechanism because the hormone itself is not triggering some secondary messenger which is going and then causing some whatever signal amplification or something. The hormone itself is going into the cell, it's diffusing through the membrane, it's moving around in this into the cytoplasm, it's binding its receptors, and then this complex is moving to the nucleus. So the hormone itself is going to the level of the gene and increasing the gene transcription rates, or it's regulating the gene transcription of certain glucocorticoid response elements. So this is a slower effect, but it's a sustained effect essentially. So this intracellular receptor mechanism of cortisol is slower, but effects last for longer. They're more sustained, right? So I hope that's clear. Cool. A few more questions on the topic of glucocorticoids in the adrenal, and then we can move on to the recap part of today's lecture. So, right, so these GRs that we were talking about, initially, what are they bound to? Does anyone else want to try? So for those of you who join later, you can send the answer that you think is the correct one, like just the option, like option A, B, C, like that, you can send it in the chat box. Um, and then we'll discuss why or why not that is the right answer. And if you have any questions, please feel free to send a question or unmute and ask me. Right. So good. I have one correct answer here. If anyone wants to contradict, if anyone thinks it should be something else, let me know. So yeah, the correct answer here is option C, which is HSP, which is a heat shock protein. So these GRs that are present in the cytoplasm, initially when they're in their inactive state, they're actually bound to these heat shock proteins or HSPs. So what happens is these HSPs essentially stabilize the GRs, the receptors, and they prevent them from prematurely being activated. And when cortisol then enters and then it binds, the HSP just dissociates from this complex, and that allows receptor activation. So until cortisol binds, HSP will prevent it from being activated prematurely. And the receptor then obviously then dimerizes and moves to the nucleus, and then all of the rest of the steps, gene transcription and so on, will happen. So HSPs are very important for binding and stabilizing the glucocorticoid receptors in the cytoplasm to prevent premature activation. Okay, all right. Let's see one more question. So what is one major long-term effect of cortisol on the immune system? So we covered this just a few questions ago. Anyone want to try? What happens to the immune system? Yeah. So I've got a few different options here. So we'll have to see which one is the right one. Does anyone else want to try answering question number 32? Yeah. So the correct answer is option C. Okay. So it's immune suppression. So I can explain why so that it's clear for everybody. So basically, what is the job of cortisol, right? The main job is that it should prioritize survival during a stress. So it needs to shift energy away from inflammation and immune activity. And because those are quite expensive with respect to energy and they could be potentially damaging if they're overactive. So that's why, I mean, cortisol is needed to sort of reprogram gene expression to reduce the expression of things like cytokines, right, which are involved in inflammation, inhibit immune cell functions, block various inflammatory pathways, and make sure that energy is being used or conserved for the things needed during that stress and inflammation doesn't become too excessive, right? So it can have genomic effects, right, where it will turn off all of these pro-inflammatory genes like NF-kappa B and the interleukins, interferons, so on. And it can turn on, oh yeah, it can also inhibit phospholipase 2, I believe, phospholipase E2. So that can block prostaglandin synthesis, leukotriene synthesis, and so on. And it also reduces proliferation of immune cells, blocks them from trafficking, so it reduces adhesion of the cells so they don't uh, exit the blood vessel as much and don't move into the tissue as much, so they can't reach sites of infection and injury too much, so that overactive inflammation is not happening. So, and direct suppression also happens. So they can cortisol can cause vasodilation to decrease. So you have essentially less blood reaching certain tissues. It can decrease capillary permeability, so you have less cells leaking out of capillaries. It can decrease edema. So these are all some like anti-inflammatory effects that cortisol can have. And it's very important to prevent overactivation of immune responses that could be damaging to tissues under a certain stress. So I hope it's clear now why cortisol has an immune suppressive effect on the immune system. Okay, it's a long-term effect, uh, and there is now, slowly coming up because a lot of behavioral research is quite lacking when it comes to controls and like effective ways to like reduce bias. But now people are looking at ways in which stress is influencing all sorts of different things in our body, right? Like if someone has a lot of stress, then it can influence their physiology in many different ways, and one of the things that it could affect is your immune system, and one of these players in this arena is cortisol, right? Along with prostaglandins, that's something also people are looking at a lot recently. Okay, last question relating to glucocorticoids. Which transcription factor involved in immune activation is inhibited by cortisol? What do you guys think? So, do you guys want to try? Great. I'm getting some answers. Does anyone else want to contest this? Or if you agree with this answer, you can also send. Yeah, awesome. So I've got some answers here. Most of them are saying NF kappa B, and that is correct. So that's option B. That's the correct answer, NF kappa B. So it's sort of linked to the fact that immune activation is inhibited. So NF kappa B, which is a very important transcription factor in the immune system, it's particularly involved in immune reactions and inflammatory responses, right? So normally what happens is that NF kappa B as a transcription factor, it enters into the nucleus and it promotes a lot of cytokine production. So cytokines are these special molecules that essentially trigger a lot of immune activation and immune cell functions, right? They trigger proliferation, they trigger immune cell activation, they trigger immune cell cytotoxicity, clonal proliferation, antibody production, all of these things, many immune functions are stimulated by cytokines, right? And NF kappa B is a very important transcription factor that promotes cytokine production when it's there. So NF kappa B moves into the nucleus, promotes cytokine production, but cortisol wants to do the opposite of all of this, right? It wants to suppress the immune system. So it inhibits NF kappa B from going into the nucleus and it prevents its action altogether. Um, and so that reduces inflammation and that reduces immune activation. So, yeah, one of the major axes of control by cortisol for this function is inhibiting NF kappa B. Okay, so I hope that is clear. All right. Okay. Great. So now we have officially sort of got through the major course materials. And so now from this class and the next, we'll do a lot of recapping. Also, just to let you guys know, I'm taking an extra class on 9th April for about an hour or so, and I'll be talking about the immune system. So I work on the immune system in my PhD work. I work on T cells. So I thought that that is a slightly less explored system as a part of this course. So I thought I'd tell you guys a little bit about it. So if you're interested, please join. I believe the class will be from 7:30. I'll have to check. I think it's 7:30. And just for about an hour or so. So it would be nice if you guys could join. Then I'll, tell you about the immune system a little bit, show you some cool videos, and that might be fun for everyone. Okay. Cool. So let's start with the recap. Now that you guys know this. Okay. So let's start from the beginning, and we'll look at some questions on the way that explain the key concepts that we've learned so far, right? Um, So homeostasis, right? We learned about homeostasis in the very beginning, actually. And essentially, what is homeostasis? It's a property of the system where, despite major or minor external environmental changes, the system is able to maintain internal conditions without too much variation. So it's able to equilibrate despite changes externally. And this is very important for maintaining an equilibrium in the body during day-to-day activities. Or if someone is adapting to some unusual environmental condition, you don't want major upset in your balances inside your body. So if you're, say, hiking up a mountain, your body finds a way to equilibrate changes in your heart rate, in your body temperature, the fact that you're losing water by sweating, and these kind of different things that are not normal to you. Your body will still find a way to maintain internal conditions with a little bit of plus minus in certain parameters. So in this way, the body would maintain homeostasis with respect to temperature, pressure, osmolarity, blood sugar, and other properties. Does someone have a question? No, I guess not. Okay. So, yeah, and we also talked about circadian rhythm, which is a very important part of homeostasis because it is an extremely important natural cycle that all of us are all, regulated by. All of us live by our circadian rhythm. If it gets upset, we don't feel good, right? So it's a natural cycle of physical, mental, and behavioral changes. Um, that the body goes through in a 24-hour cycle. And these rhythms are mostly affected by light and darkness. And they're controlled by a small area in the brain. And there are some very important genes called the clock genes that are part of regulating your circadian rhythm or your circadian clock, right? And these can affect your sleep, your body temperature, your hormones, like serotonin, melatonin, all of these are very important in regulating your circadian rhythm and your diurnal pattern. So, yeah, and you can have seasonal affective disorders, sleep disorders such as insomnia, people who work late nights or work night shifts in their jobs, they can have a lot of circadian rhythm related problems. So they can suffer from physical, mental, or behavioral issues because they're just not able to regulate their rhythm properly, right? So the body's clock is upset, and so that can lead to health issues, right? And as I mentioned, that there are some important clock genes that are part of using these positive and negative molecular feedback loops. Um, and these genes essentially regulate and control transcription and translation of various important factors. And yeah, expression of these are very important to identify because the body I mean, aside from your rod cells in your eyes essentially telling about dark or light, there's no other real way to identify time of day and perform a program function. So actually, there are these negative and positive molecular feedback loops that allow for this to occur. Great. Right. So there's a question here that I designed, which you guys can read, and then we can go through the options. So let's look at this question. It's related to what we just talked about homeostasis and so on. So let's read the question, right? So a researcher studies two groups of individuals exposed to different lighting conditions. So group one experienced a constant dim light, while group two underwent an 8-hour phase advance. So basically, simulating eastward travel. So after several days, group one maintained a free running rhythm of about 24 and a half hours. So note this. While group 2 showed disrupted sleep, hormone secretion, and body's temperature cycles all were upset. So now, which of these following options would best explain the physiological differences that occurred between these two groups? So the first option is that group 1 has impaired suprachiasmatic nucleus signaling and group 2 has normal melatonin secretion. Option B is that group 1 shows endogenous circadian rhythmicity independent of the external cues that it experienced, while group 2 experiences circadian misalignment, so basically an upset in circadian cycle with environmental Z-givers. And C is a group 1 has increased cortisol amplitude, so higher cortisol, while group 2 has decreased basal metabolic rate, if you guys remember what this means. And finally, group 1 lacks peripheral clock gene expression altogether, while group 2 has enhanced synchronization of central and peripheral clocks. So this is a bit of a thinker. So you guys think and let me know what you think the answer is. So for those who may have forgotten, an environmental Z-cabo is essentially an external cue that helps to synchronize the biological clock. Okay. So if you remember about how this synchronization happens, like what anatomical component is there that helps regulate this, you might be able to sort of easier tell the answer. Yeah. So I'm seeing some answers. Does anyone else want to try before I explain? Okay. So the correct answer here is option B. Okay. So that is where group 1 demonstrates an endogenous circadian rhythmicity that's independent of external cues, while group 2 experiences circadian misalignment with environmental Z givers. So what does this mean? So let's break it down. Group 1 has essentially a free running circadian rhythm. That means that the internal clock, which is, by the way, the internal clock is driven by this thing, suprachiasmatic nucleus or SCN. right? So in group one, who were exposed to a constant dim light, meaning there was no sort of light ones and then dark ones. So this cycling was not happening. They constantly had dim light, right? They did not have any external cues of light. But group two had a circadian misalignment. Why? Because there was a phase shift. What does this phase shift really mean? It just means it's like a jet lag, where the internal rhythms essentially became out of sync with the environmental Z givers. And as I mentioned, a Z giver is essentially um, it's like a external cue that can influence the um, that can synchronize and influence the internal biological clock or your circadian rhythm. And this is via the suprachiasmatic nucleus and the hypothalamus. So the Z givers essentially are to some degree communicated to the SCN, which then helps to align your internal biological clock with external cues. And light is one of the most powerful environmental Z keepers. Um, so it's like morning light essentially resets the clock and promotes your wakefulness, and night light suppresses melatonin, delays your sleep. Um, so yeah, so basically, where were we? So yeah, we were looking at how group two essentially experienced a circadian misalignment because there was a phase shift. So essentially, the internal rhythms were out of sync with light cues. So because they advanced 8 hours, meaning that the light cues that normally one would have at say 8 a.m. in the morning was advanced by 8 hours. So it's like you're moving. What happens if you travel east, right? It's like you're moving ahead, right? Suppose you're living in India and you move to Japan. So Japan is ahead of you in time. It's towards the east. So it's like you are having suddenly um your sunrise is like some X number of hours before what you're used to, right? So this 8 hour phase advance basically means your sunrise is happening 8 hours before your body is used to, right? Within that period of time. Obviously, everyone can, if you shift to Japan permanently, your body gets used to it. And then if you move back to India, you'll have a some kind of jet lag or phase shift. So this kind of jet lag can cause disruption in um, in sleep hormones, in sleep hormone secretion like melatonin, like cortisol levels, and the temperature also can be misaligned because the clock itself is failing. So you all of these different cues, like normally your body is a little colder when you're sleeping, these things will all be out of sync. So that's why the answer here is option B. Right, so I hope this is clear. Yeah. Okay, so let's move to the next question, if this is fine for everybody. Okay, so there's a patient who is working night shifts, rotating night shifts, and they are saying that they have chronic fatigue. So they're really tired all the time, basically. They have impaired glucose tolerance and increased blood pressure. Okay, so all bad things. Despite maintaining consistent caloric intake, meaning they're eating consistently, and sleep duration, meaning they're sleeping consistently, the length of their sleep is consistent, these metabolic disturbances persist regardless. So, which of these mechanisms could accurately explain why this is happening to this person? So, these are the options. Increased parasympathetic tone during nighttime activity enhances insulin sensitivity. Disruption of your peripheral clock synchronization leads to some mismatch between metabolic enzyme activity and nutrient intake. There could be elevated melatonin levels during daytime sleep, which directly cause gluconeogenesis because of cortisol. And finally, you can have reduced amplitude of circadian oscillations, which eliminates hormonal feedback regulation. So, what do you guys think? Which one is the correct answer? Take your time and think about it and let me know what you guys think is the correct answer. For those who joined late and stuff, like basically, this is the question. We're going through it, and whatever you think is the correct answer, you can send that option. Like you can say option A, B, C, whatever, in the chat box, and then we'll discuss it after a reasonable number of you have tried answering it. Cool. I think this one has successfully befuddled you guys. So I'll explain it to you. But I'm sure you'll get it very easily. So, as I mentioned, with the respect to the circadian rhythm, right? Sleeping at the correct time, eating at the correct time, these are all very important factors um, with respect to maintaining circadian rhythm, right? So now if someone is working a night shift, a rotating night shift, that means that there are nights when they don't sleep, right? So those nights, their circadian rhythm is going to go all over the place. So shift work causes desynchronization between your suprachiasmatic nucleus, so your which is very important for regulating your circadian rhythm, and peripheral clocks, like in the liver and the pancreas, right? Liver for gluconeogenesis, for instance, pancreas for secretion of insulin, for instance. So even if sleep duration and caloric intake, as mentioned, right? That sleep duration, how long they slept, and caloric intake, how much they ate, these are stable. The timing mismatch between food intake and metabolic enzyme activity can impair how your glucose handling happens and increase all these risks like cardiometabolic risks and so on, right? So the if you eat at a certain time but your enzymes are not present or not active, then that's going to cause, food to not be digested, sugar to not be broken down. All of these mismatches can happen just because there's a timing mismatch, right? So this reflects how there's, a very important breakdown in circadian coordination when it comes to physiological homeostasis. And it's not simply just changes in hormone levels or some autonomic tone. Um, so yeah, so the answer here is option B. I think I forgot to mention that, right? So because they are sleeping at the wrong time or they're not able to sleep at the proper time, that can cause peripheral clock synchronization disruption. So peripheral clock and linked to your suprachiasmatic nucleus, of course, regulation, and that leads to a mismatch between when your enzymes are metabolically active and when you are eating, for instance. So these disturbances will persist unless that person does some way of, some repercussion, some takes maybe some medicines or regulates by, changing out the way that their night shifts work. Cool. Right. So that was about homeostasis. Now we come to a very important topic, which is, of course, the skeletal muscles, which was the next thing covered in the coursework and has a very important bearing with respect to, um, I think it'll have quite a lot of weightage in the final test that you guys give. So I think most of you guys would remember because this was covered in quite extensive detail, but let's just go through it real quick, right? So I think I'll put a, yeah, cool. So, right. So what we're looking at over here is the skeletal muscle, right? And we're looking at essentially the hierarchical organization of the skeletal muscles going from outside to inside. And so there's this multi-level structure that the skeletal muscles have. So you start with the whole muscle fiber, right, or the muscle cell, and it is a single long multinucleated cell, and it's surrounded by your it's surrounded by the sarcolemma, And it has multiple nuclei, it has lots of mitochondria for energy and ATP production, it has myofibrils, which are the contractile units. So you start on the outside with this sarcolemma, nucleus, mitochondria, right, myofibrils. And then you break down the myofibrils further. And if you look at one long myofibril, what do you see, right? You have these repeating units called sarcomeres. And that's why these skeletal muscles are called striated muscles, because they have this stripy appearance, right? So the sarcomere is the functional unit of the skeletal muscle, and it's the smallest unit that's capable of contraction. So what are the boundaries of this sarcomere, right? So it is the Z line, or the Z disc, as people call them. Right, so there's a Z disc at either end. So here in this one, it's easier to see, right? So you have Z disc here and another one here. And these are the boundaries of the sarcomere. And so what are some important regions inside this repeating unit? So you have the dark band, which is your A band, right? Or anisotropic band, right? So this has the thick filaments or the myosin filaments, right? And this is the region where there's an overlap between myosin and actin. As you can see, the green is actin, right? Or the thin filament. So the thick filament is myosin, and that's over here in blue. The thin filament is actin over here in green. And in this A band is where this overlap takes place up until the H zone where the overlap stops, and it's only the thick band or only myosin. Now, if you move outwards, you hit the Z disc. At the Z disc, um, you have the I band over here, right? The I band extends on either side of the Z disc, and this is the isotropic band, and it only contains thin filaments or only contains actin. Okay, and it extends from the Z disc to the start of the myosin over here. So during contraction, the I band is what is actually contracting. And then here you saw I mentioned the H zone, right? So that has only myosin. There's no overlap with actin over here. Um, yeah, and during contraction, it actually kind of disappears. And then you have one last thing, it's mentioned here, which is the M line. So the M line is in the middle of the sarcomere, and it actually holds the myosin filaments together. Right? So it's important for holding them together. So the M line is sort of in the middle of this, right? Cool. Uh, yeah, so this is sort of a little more detailed version of this where you have some more accessory proteins that are seen. Titin sort of goes the extent and holds all of this together. Alpha-actinin helps with anchoring. All these different proteins have many different functions. We won't go into them in too much detail. Yeah. Okay. Apparently that's all I had for this. Does anyone have any questions about any of this? Then I'll move on to the sliding filament theory, and then I think we have some questions. Yeah. So over here, I think I forgot to mention, I mean, I can quickly just recap this. Essentially, the thin filament of actin, right? What all is there, right? So you have the thin filament containing actin, then you also have tropomyosin and the troponin complex, which we'll come to when we're talking about the sliding filament theory. And then the thick filament of myosin, that has the globular myosin heads, right? Which bind actin and they hydrolyze ATP. And that's what forms these cross bridges during contraction. These cells also, alongside mitochondria, they also have sarcoplasmic reticulum, as seen here. And this is a very specialized endoplasmic reticulum, essentially, that stores a lot of calcium ions and releases calcium into the cytoplasm, which triggers contraction. So essentially, they're like a calcium reservoir, right? And so then we'll talk about the sliding filament mechanism. So what happens to the actin and myosin that allows muscle contraction. So what is the sliding filament theory, right? It's a very important one, and it talks about how actin, which are the thin filaments, and myosin, which are the thick filaments, actually slide against each other to facilitate skeletal muscle contraction. So when a muscle is stimulated, so there's these neuromuscular junctions that allow for stimulation of a muscle such that there is contraction. So if a muscle is stimulated, calcium ions are released, like a whole bunch of them are released from the sarcoplasmic reticulum. Now, if you guys remember about troponins, right? So troponins are these important proteins found at this site, and particularly one type of troponin, which is troponin C, is the one that is sort of blocking a certain site. And what these calciums that are released from the sarcoplasmic reticulum do is they bind troponin C and they cause a sort of conformational shift that moves tropomyosin, which is also there bound next to troponin. It moves tropomyosin away because it shifts in its conformation. And it moves away from the myosin binding sites of actin. So those are blocked initially, and this calcium hence helps to unblock those sites. And that is what then essentially allows the myosin heads to be energized because they are already like bound to ADP and phosphate, and then because like before when there's ATP hydrolysis, so then this allows these myosin heads to then get exposed and then bind to the exposed sites of actin and form cross bridges, right? So where are we? Yeah. So you have myosin, it hydrolyzes ATP. I guess we can essentially start probably from here. Yeah, so the cross bridge forms between myosin and actin with the help of ATP hydrolysis. And then what happens is myosin performs this pulling action. So this is what's called a power stroke. Okay. And releasing this phosphate was what triggers the power stroke. And so then the myosin head sort of pivots this way and pulls the actin filament towards the center of the sarcomere, which is the M line, right? And then the ADP is also released. And then a new ATP molecule, which is here, it comes and binds to the myosin head and causes it to detach. Because when ATP is bound, it does not, the myosin head is not attached to actin. So it detaches, right? And then you can go into a subsequent ATP hydrolysis mode, and then you reposition and get the myosin head energized in its high energy state all over again and ready for the next cycle. And so this is repeated again and again, and it doesn't necessarily have to occur synchronously across many cross bridges across the muscle, and that results in shortening of the sarcomere. So the Z discs essentially move closer to each other. The I bands and the edge zones shrink, and the A band actually remains constant. And this is what is important for generating muscle contraction. So this will only happen as long as ATP is available and calcium is available that's coming out of the SR, right? So as I think this was mentioned in the coursework, but if you guys remember about rigor mortis, so this is something that occurs a couple hours after death, right? So this happens because ATP is depleted in the body because the body is like dead. So it's not producing energy anymore. So ATP is depleted. So the myosin heads can't detach from actin because only when ATP binds is the myosin head able to detach. So it's sort of stuck to actin and in a stiff, like unmoving position. And that's why bodies go into rigor and you can't move their muscles very easily. Yeah. So a single muscle contraction like this actually involves actually billions of cross-bridge cycles that happen at the same time. Not necessarily synchronously, they can happen asynchronously. Yeah. So yeah, calcium is super important in this regard. Yeah. So does anyone have any questions regarding this mechanism? I can go into the next question and then we can sort of move on. Okay. So imagine that there is a mutation that alters the Z disc anchoring proteins in the skeletal muscle fibers. The Z disc anchoring proteins. Okay. And this is causing weakening of the attachment of actin filaments without directly affecting myosin or ATP availability. So during contraction of the skeletal muscle, which of the following outcomes is the most likely? So these are the options. I won't bore you by reading them out. Read through them and think about what the correct answer might be. So let me know what you guys think. To anyone who doesn't know, you can read this question, think about which is the correct option, and send that into the chat, and we will discuss. Does anyone want to try? So I've got one answer so far. Does anyone else want to give it a shot? Okay. So the correct answer here. Oh, okay, okay. I'm getting some more answers. Hold on. Okay, so I have a 50% saying A, 50% people saying C. I wonder. Does anyone else want to try or, contribute to this tiebreaker? Someone has raised their hand. Do you have a question? Okay. All right. So great. So the correct answer, I will put you guys out of your misery. The correct answer is option A, okay, which is reduced force transmission despite normal crossbred cycling. So why is that, right? So the Z disc, right, as mentioned here, it anchors actin filaments and it's important for transmitting force across sarcomeres, and ultimately that will go and whatever touch to the tendon and so on. So now if this anchor at the Z disc is weakening, cross-bridge cycling can still occur, right? It can still occur normally since myosin, actin, calcium, ATP, all of these major effectors in cross-bridge cycling are unaffected. So your cross-bridge cycling will still occur. But the amount of force generated cannot be efficiently transmitted along the muscle fiber. That's why you will have a reduced effective sort of force output because of this. So this sort of there's a distinction here between force generation and force transmission, because the force is generated, it needs to move along the muscle fiber and eventually reach to the tendon or wherever. So it's sort of subtle, but it's important. Um, so that's what's happening over here. You have a weakening of the attachment. So actin, myosin, ATP, calcium, everyone's still there. So cross-bridge cycling is still happening, but your force is not transmitted. Does this make sense? Right? You can see here, right, that um, I don't have that image unfortunately, but yeah, you have anchoring here at the Z disc. But if this and these guys, all of these guys are there at the Z disc, right? That help with this anchoring. If this is becoming weak, then the force will not be forward properly to the next one and the next one. So this transmission will not happen adequately. Great. Uh, right. So I hope that's clear. So we can move to the next question. Right. So, okay. So here is another hypothetical sort of experiment where intracellular calcium levels were artificially elevated, maybe using some drug or something, in a skeletal muscle fiber, but troponin C was made non-functional. So what could likely be the consequence in this case? So do you guys want to try? You can answer in the chat box. If you are, even if you're not confident, it's totally cool. Just give it a shot. Yeah. So I've got some answers here. Good. Great. I'm getting lots of answers. So I think an overwhelming majority have said option B, and you guys are absolutely right. It is option B. So why is it option B, right? So troponin C, as we had discussed a little earlier, is a calcium binding subunit, right? That is important for initiating contraction because it shifts tropomyosin away from the myosin binding head site on actin. And that's because essentially calcium binds to troponin C, and that triggers conformational changes that allow it to shift tropomyosin away. Now, if troponin C is not functional, meaning in this case, I know it's a little bit confusing. It's like, is it non-functional in the sense that it's not blocking the site, or is it like basically by non-functional, I meant that it does not bind to calcium? So if troponin C is non-functional in the sense that it can't bind calcium anymore, despite even like a high level of calcium, in this case, it's been artificially elevated by maybe some drug or some treatment, right? So even if you increase intracellular calcium like anything, because troponin C has a problem, it's dysfunctional, it does not bind to the calcium. So then troponin C cannot move tropomyosin, so the binding sites between myosin and actin remain blocked. And because of that, cross bridge formation will not happen and contraction will basically not occur. And so calcium alone is insufficient because troponin itself, troponin C subunit specifically, needs to be active for this to occur. So if there is a mutation, for instance, or some loss of function mutation in troponin C, then and that prevents active sites binding to calcium from functioning, then that could be that could be really um, very potentially problematic. Yeah. Cool. All right. Great. So good job to all of those who got it right, and to those who didn't, I hope it's clear now. All right. Let's look at the next question. So a pharmacological agent, this is just another hypothetical. So this selectively stabilizes tropomyosin in its blocking position on actin. Okay. So we're just talking about this, right? Even in the presence of calcium, tropomyosin is still in this blocking position due to this particular pharmacological agent, or basically this particular drug. So which of the following would best describe the expected effect on muscle physiology from this case? So it's sort of similar to the previous question. Okay. So I think, in interest of time, I'll go through the answer now. But I'm seeing people have answered option B, and you guys are absolutely right. It is option B. So, which is myosin binding to actin is prevented, and this reduces force generation. So, why is this the correct answer? Because tropomyosin, as we know, it is physically blocking the myosin binding sites on actin in the resting muscle, right? So now if we artificially just forcefully sort of hold it in position and keep making it block that position, myosin heads cannot attach to actin, right? And even if calcium is present and troponin is activated, regardless of all of that, we are using some drug that's overriding this entire system. And so myosin to binding to actin is prevented and force generation is reduced. So regardless of everything that's needed, everything that's present, if we block any one of those basic steps, then uh, like the power stroke will not occur and forces will not be produced. So basically, the takeaway from this is that the most important sort of players at the cross bridge cycle is obviously calcium and ATP, and also troponin, tropomyosin, as well as obviously actin and myosin itself. Right, so those are the important ingredients in the recipe for cross bridge formation and cycling. All right, one more question and then I think we'll move to a different topic. So, yeah, a researcher compares two skeletal muscle fibers. So, fiber X has longer sarcomeres stretched beyond optimal overlap. So, if you guys remember the overlapping positions, right? And while fiber Y has very short sarcomeres and that has excessive filament overlap. Both are stimulated maximally, so some external sort of in vitro stimulation. What is the most accurate comparison of their force generation? So read the options and let me know what you think is the correct answer. What do you guys think? Great. So I'm getting lots of answers. Happy for that. Nice. Okay. Great job. So you guys are right. It is option D. Yes, great. So it is basically force generation depends very critically on having an optimal overlap, right, between actin and myosin filaments. So now in the case of fiber X, if you have like overly stretched sarcomeres, then fewer cross bridges would be able to form due to reduced overlap, and so you would not have uh, proper amount of force generated. And the in the flip side, if you have the opposite condition, which is overly compressed sarcomeres, like in fiber Y, right, then your filaments would overlap too much. And that would cause interference between them and actually reduce effective cross-bridge formation. So there is an optimal sort of Goldilocks zone, which needs proper, which allows for proper force production, And despite however much stimulation you give, if that is suboptimal, then you will not have the correct amount of force generated. And so this length tension relationship is very, very critical to skeletal muscle contraction and muscle physiology. All right, great. Now, oh, excuse me. Yeah, so now we finished striated muscles. We talked about some basic concepts. If anyone has questions, please let me know. We can move on to cardiac muscle structure now. So, yeah. So this, you pretty much know, cardiac muscles themselves, or cardiomyocytes, they look a little bit like the skeletal muscles. They're striated, they're branched, they have lots and lots of mitochondria, understandably so, and they are under involuntary control. It would be catastrophic if they were under voluntary control and you forgot to make your heart beat. So each myocyte contains a single centrally located nucleus. So that's different than the skeletal muscles, right? Which is like this, the skeletal muscles are like a syncytium. So here it's surrounded, it has one nucleus surrounded by the sarcolemma, and they are, these cells are connected though to one another by these intercalated discs. So they have these branches in between that are connected via intercalated discs, like you can see over here. And these discs contain gap junctions, adherent junctions, and desmosomes, very important components that help with information transfer between the cardiomyocytes. And these interconnections are important for um, the transfer of mechanical forces, electrical information, and this allows cardiomyocytes to contract synchronously, because regulation of the heartbeat, making sure that things happen synchronously, is what allows the heartbeat to happen in a rhythmic fashion. So rhythmicity of the heartbeat is is highly influenced by the ability of cardiac muscle cells to communicate via these intercalated discs to one another. Yeah, and importantly, the sarcolemma, so the cell membrane of cardiac muscle cells, contain voltage gated calcium channels. So they have these ion channels that are different from skeletal muscle cells. They depend highly on their sarcoplasmic reticulum, whereas here you have a lot of calcium coming in from outside. And you also have these T tubules or transverse tubules, and they are very branched invaginations into the cardiomyocyte sarcolemma. And they are very important in excitation contraction coupling. Essentially, um, when there is an excitatory stimulus and then there is contraction of the heart, um, and this the regulation and rhythmicity of that. T tubules are very important when it comes to the entry of ions for that process. Then we had looked at in previous discussion sessions about the electrical regulation of the heartbeat, right? So you have these phases defined for when you have depolarization, repolarization, and then eventually there's like a resting, and then it goes again. And basically every heartbeat, this entire cycle repeats. So at the start, you have phase zero, where you have rapid depolarization. So at resting membrane potential, we are at about minus 85 millivolts with respect to the inside of the cell. And then you have these voltage gated sodium channels opening, and these are really fast, okay? Fast channels. And they allow this rapid influx of sodium ions. So the inside was negative, and now you have a whole horde of sodium ions entering, which are positive. And because of this influx of this cation, sodium, right, the membrane potential changes all the way from minus 85 millivolts to plus 35 millivolts, right? And so these sodium channels, they're called fast channels, right? So they're really quick, they open really quick, they close really quick. And that's why there's this very steep upstroke of the action potential. And this is different from the calcium channels that are slower. So then you have phase one, okay, so which is an initial repolarization phase. So now your voltage gated sodium channels have closed, so there's no more sodium coming in. And you have these potassium channels that start to open. And there's a slight like the beginning of a slight outward potassium current. And because of that, there's a very slight drop in electrochemical potential. That's here. And that is moving into phase 2, which is the plateau phase, where you have your calcium influx starting. So these voltage gated L-type calcium channels, which are much slower than these fast sodium channels, right? They open and calcium influx begins, and that actually balances out the K plus that's effluxing already. And because you have calcium coming in, potassium going out, you have a plateau because it is sort of balancing out efflux and influx, right? And this plateau is very important. It's a component of the effective refractory period, where essentially the influx of calcium also stimulates calcium release from the sarcoplasmic reticulum, and that initiates muscle contraction. So there's a two-phase calcium-based regulation, right? So you have calcium coming in from outside through your T tubules and so on into the cytoplasm, and then that, I haven't gone into detail there, but we had talked about it in earlier lessons, where essentially calcium comes in from outside, and then that calcium in the cytoplasm further triggers more calcium release from the sarcoplasmic reticulum, and that ultimately allows for optimal muscle contraction. And then the last phases, which are the repolarization until you go back to baseline. This is basically where K plus efflux through this rectifier K plus channels continues, and voltage gated calcium channels close. So there's this slow leakage of potassium that's going on, which is why you continue to repolarize very rapidly. And then finally, you reach resting membrane potential. And then the whole cycle continues all over again. Right. So this electrical regulation ties in nicely with the heart rhythm, where you have your different waves that you will see in an ECG, which is an electrocardiogram, right? Where you have leads put on a person's body at different parts of their body. And this gives you essentially this shape of a curve that tells you the length, duration, and rhythmicity of the different contraction and relaxation events that occur in the heart. So we'll start here with the P wave. So the P wave is when atrial depolarization is occurring. So the mechanical event here, so essentially I've divided it here. So you have an electrical event, which is atrial depolarization. This basically means it's like, you can sort of think of it like activation. So depolarization means contraction. So you have atria here, left and right atria, that are depolarizing, meaning that this is happening here. Depolarization means this initial shooting up phase. Sodium channels are open. And you have atrial contraction. Then, so what does atrial contraction mean? The blood is moving from the atria, being pushed from the atria into the ventricles. That's why the AV valves are open, atrioventricle valves are open. Then you have PR segment, so between P and R. So that's just a slight delay of the AV node. During this time, the ventricles are filling with blood that is being pushed into it by the right and left atrium, and the AV valves are still open. Then you have this peak, which is the QRS segment or the QRS complex. That is when ventricular depolarization occurs, meaning the ventricles, now they're fully filled with blood, ventricular contraction begins. So the AV valves now need to close, otherwise blood would flow back into the atria. So the AV valves are closed and the ventricles are going to start contracting. Then you have the ST segment where your ventricles are fully depolarized and the ventricles eject the blood into the aorta and into the pulmonary vein. Yeah, and the AV valves are closed at this time. And the semilunar valves, which are the ones that are guarding these two blood vessels, those are open. And finally, you have the T wave, this last little bump, where ventricular repolarization has occurred, meaning that now it's time for ventricles to relax because the blood has been pumped out. And the semilunar valves close to prevent backflush of blood from, say, the aorta, for instance. and then we get ready for one full new wave of this, and this is occurring constantly in about zero point 0.8 seconds. So that is the heart rhythm. That's an ECG. These are the components that you see in ECG. This is a healthy ECG, right? This is how it should look. But there can be pathogenic conditions where someone is having, tachycardia, bradycardia, where this thing will be really fast or really slow, respectively. ST elevation can indicate an incoming major heart event, right? And oh yeah, we talked about this, but essentially electrical system of the heart, right? So there are some external nervous inputs, and then you have the heart's own regulatory mechanism starting from the sinoatrial node, which transmits electrical activity to the AV node or the atrioventricular node, which then goes via the bundles of His and Purkinje fibers to the ventricular muscles, and spreads this electrical information. And this slight delay is important between SA and AV node, which allows for the proper regulation of first atrial systole and then ventricular systole, right? Or contraction, basically. Right. So now with that recap, hopefully whatever you guys remember about heart stuff is now coming back to you. Let's look at this question. So there's a genetic defect, let's say, right, that weakens desmosomal proteins in cardiac muscle cells, but it doesn't affect the gap junctions. So which of the following is the most likely physiological consequence when there are these repeated cardiac cycles? So let me know what you guys think is the answer. Anyone willing to try? Okay, I have a few answers here. Great, I'm getting lots of answers. I think all of you are on this correct uh, thought process here. Yeah, everyone has answered B, which is great because that is the correct answer. So desmosomes, as you guys must remember clearly, are mechanical junctions. So they are what hold cardiac muscle cells together. And during this, pretty intense mechanical stress of repeated contractions, desmosomal integrity is really important. So if it is compromised, then the cells can actually detach from one another, right? And so you have reduced mechanical cohesion due to cell detachment during contraction. That's the correct answer. However, electrical conduction would remain intact because gap junctions are what are important for communicating electrical information across the different cardiac muscle cells. And so if the gap junctions are not affected by this genetic defect, whatever it is, then electrical conduction will still be the same. So you still have high electrical coupling, but that is actually a problem because then you have mechanical wear and tear because the gap junctions are already weak, so the cells might actually come apart, which is as horrible as it sounds. Right. All right. Let's do a few more questions and then, yeah, I'll most likely try to wrap up. Okay, here's another question. So a drug selectively blocks fast voltage gated sodium channels in ventricular cardiomyocytes. Which phase of the cardiac action potential is most directly affected and what is the expected outcome? So we just talked about these phases, right? So let me know if you guys remember, what do you guys think? Great. I'm seeing a lot of answers. Yeah. Awesome. Yeah. I think all of you are on the right track with this. It's option B, which is phase zero. Uh, so you'd have a decreased rate of depolarization and slowed conduction velocity. Because in phase zero, what's happening is that the ventricular action potential is driven by the rapid sodium influx through the fast voltage gated sodium channels, right? And so that is what that steep depolarization jump is initially from minus 85 to plus 35 millivolts. And so blocking these channels would reduce that phase, the slope of that phase entirely. And that would slow down conduction velocity across the myocardium. And so that's why, when patients come with like some arrhythmia problems, so some irregular heartbeat issues, sodium channel blockers are used um, as basically an antiarrhythmic agent, which can reduce excitability and regulate the heartbeat better. So I hope that's clear to everybody. Let's move on to the next question. Okay, so this is about your ECG. So a patient's ECG shows a prolonged PR interval, but otherwise everything else is normal. So which of the following best explanations could talk about what could be the underlying physiological alteration here? Do others want to try? I'm getting some answers here. Great. Yeah, so you guys are right. It is option C, which is that there's a slowed conduction through the AV node. So how does one explain this answer, right? So the PR interval, okay, this PR interval is representative of the time from atrial depolarization, so when the atria are contracting, to ventricular depolarization, right? So this includes the delay at the AV node, which is necessary for making sure that the atria and the ventricles don't contract at the same time. So if there is a prolonged PR interval, what does that mean? It means that the necessary conduction between SA node to AV node to initiate ventricular depolarization, that has got slow. So normally there is a certain amount of delay that allows ventricular filling, but if it's even longer, that means that there is a slower conduction through the AV node for whatever reason. But since everything else is normal, it means that, I mean, The issue is localized to just nodal conduction rather than the entire ventricular depolarization repolarization. That is happening. It's just slower. So that, it's a problem in itself. Okay. All right. Let's do one more question on this topic, and then I think I have like one last thing to cover, and then we're done for today. Okay. So question 10. So during an experiment, calcium channel activity during the plateau phase, which was phase 2, if you guys remember, right, of the cardiac action potential is significantly enhanced. So, what are the effects that are most likely to be seen? So, you can imagine that maybe there's some heart where some sort of in vitro experiment is being done, some cells, some tissue, or something. What do you guys think? Yeah, great. Yeah, I'm getting some answers. All correct. Good job. So it is indeed option C, which is prolonged contraction and increased force of cardiac muscle. So as we saw earlier, phase 2 or the plateau phase is maintained by a balance, right, between your calcium influx and your potassium efflux. So now if you have increased calcium channel activity, then that means you have increased calcium entry. So what would happen is this would prolong the plateau phase and enhance excitation contraction coupling. And that could lead to sort of stronger and longer lasting contractions of the muscle. And so it's kind of important, um, sort of highlights how important calcium is, right? In cardiac contractility and yeah, the fundamentals of ionotropy, right? But this is not a good thing. Just because there's like a better contraction doesn't mean it's a good thing, right? It can lead to dysregulation of the rhythmicity as well. All right, great. Uh, and this was an important pathway. I cannot cover everything, obviously, that has been sort of discussed in the course, but I thought this is a very important pathway with respect to regulation of blood pressure, right? And we were talking about it earlier when we were talking about glucocorticoids, right? And this is a renin angiotensin aldosterone pathway. So you guys must remember this. This is the RAS, basically renin angiotensin aldosterone system. So this is a very important hormonal cascade, essentially, and it maintains the blood pressure, the blood volume, and electrolyte balance. So it is in response to when there's reduced effective circulation volume. Essentially, if someone is undergoing dehydration, if there's low sodium or sodium deficiency, or there's hemorrhage, if there's an injury and there's like loss of blood, and this is all told to, like leading to decrease in blood volume. Decrease in blood volume would lead to decrease in blood pressure, right? And this drop in and decrease in blood pressure would lead to essentially decreased renal perfusion. And this is sensed by cells in the juxtaglomerular apparatus of the kidney. So the JG cells or the juxtaglomerular cells, those are the guys that sense this drop in renal perfusion in the kidney, right? And they release renin. Okay. So renin is what initiates then the conversion of um, this angiotensin that's produced by the liver, and renin initiates the conversion of angiotensinogen to angiotensin 1, right? And then angiotensin 1 is further converted in the lungs by this enzyme called ACE, which is secreted by the lungs, and that is called angiotensin converting enzyme, ACE enzyme, right? And angiotensin 1 is converted into angiotensin 2 with the help of ACE in the lungs. And then this angiotensin 2, which is now the final active form, is and a central effector molecule essentially in all of this. This is what goes and causes really potent vasoconstriction, so constricting of blood vessels, causing raise of blood pressure, right? And then it stimulates the adrenal cortex, um, and that is what secretes aldosterone, right? And that acts on the distal nephron to increase sodium and water reabsorption, and it promotes potassium secretion, excretion, and hydrogen excretion, H plus secretion. And so that expands the blood volume quite a bit. And there's a lot of feedback loops that regulate RAS. So if you once restore the blood pressure, so all of this is done, and now restore the blood pressure, it's normal again. And the volume that the increase in volume then eventually suppresses renin release. So there's a negative feedback. So once you've achieved the required blood pressure, this pressure can actually then tell the kidneys that we don't need any more renin. So then that's negatively feeding back there. And increased extracellular potassium can directly stimulate aldosterone secretion independently of renin actually. So then there's signals from the macula densa as well that sends a lot of tubular sodium chloride that can fine tune renin. So there's a lot of different regulatory factors that can essentially regulate the RAS pathway. And so you have all of these hemodynamic sensing and these hormones and these different things that are fine tuning RAS and restoring homeostasis properly, right? So with regard to this, I have, I think, two questions for you, and then we can wrap up for today. So, right. The example is a patient who has bilateral renal artery stenosis, and I'll explain what that is, develops persistent hypertension, meaning constant high blood pressure. Despite elevated systemic blood pressure, so that means in their whole body they have constantly have high blood pressure, their renin secretion remains really high. So which of the following best explains this paradox? So think back to the RAS system and let me know what you guys think. This is what bilateral renal artery stenosis is, by the way. It's essentially where there is this sort of, it's like it's stuck. So in case this is confusing, so bilateral renal artery stenosis, I might as well just tell you guys. So the kidneys are essentially receiving less perfusion pressure because the renal arteries sort of get narrowed, as you can see here. Bilateral basically means both of them. Both the kidneys are getting reduced perfusion pressure or blood entering into them because these guys have got quite narrow. So that's a sort of disease condition. So with that, now I hope you guys can answer. Okay, so I'm seeing most of you have answered option C. Option C. Does anyone else want to try? So the correct answer here, in interest of time, is option A. Okay. So there's reduced perfusion pressure at the afferent arteriole because of this stenosis, and that is sensed as low effective circulating volume. So basically, what's happening in your bilateral renal artery stenosis is that the kidneys will receive reduced perfusion pressure because the renal arteries have got narrow, and the juxtaglomerular cells, which are the cells in the kidney that are sort of getting this signal, they'll interpret this as low effective circulating volume. And even though systemic blood pressure is high, the kidney is interpreting this as low volume because less blood is being received by the kidney. And so this just triggers renin release because you have to remember the RAS pathway is sensitive to when blood pressure is low or supposedly low. So the kidney thinks blood pressure is low because less blood is reaching it, but actually that's not the case, right? So it unfortunately will then persistently release renin that activates the RAS pathway and that further increases blood pressure. And so that sort of the feedback mechanism here is just completely overridden because locally the kidney is thinking that okay, there's less blood, and that is overriding the whole system because actually that's not the case. So the kidney's own perfusion status is what is um, sort of being prioritized over what is happening in the whole system. Okay, let's move on to one more question and then we can finish for today. All right, so here is an example of a patient who is given an angiotensin converting enzyme or ACE inhibitor. And shortly after this, they experience a decrease in blood pressure and mild hyperkalemia. This is relating to potassium levels, right? So, which of the following mechanisms most directly accounts for the rise in serum potassium, which is their hyperkalemia? So, what thinking back to again this RAS mechanism, could you guys answer? Okay, so I'm seeing the answers now. I think everyone's got it. So I'll just quickly explain it and then we can wrap up. So it's option D, you guys are all right, which is decrease aldosterone levels, reduce potassium secretion by principal cells. So essentially ACE inhibitors, they reduce the formation of angiotensin 2 because they are the guys that convert angiotensin 1 to angiotensin 2. And this in turn, which will lower the aldosterone secretion from the adrenal cortex because that entire pathway is now shut off. And aldosterone, normally, what does it do? It promotes sodium reabsorption and potassium excretion in the principal cells of the collecting duct. And when aldosterone is not, the levels have decreased, right? Potassium secretion will decrease. And so the accumulation of potassium in the blood will increase. That leads to some level of hyperkalemia. And yeah, so electrolyte homeostasis, basically potassium balance, can be regulated by this thing. So if you take ACE inhibitor for the purpose that, okay, uh, you don't want your blood pressure to increase, then you might have slightly higher potassium in the serum. That's just one byproduct thing that might occur. Okay, and with that, we're done for today. Uh, Great that a lot of you have joined. So if anyone has anything that they're confused about, you can let me know now, and I can explain it if you like, or we can continue in the next class where I'll be covering further topics that were covered in past lectures, past weeks. And please, everyone, attend the extra class, which will be, I think they'll send a mail out about it. I'm not sure. Which is going to be happening on 9th at, let me just check what time it is. Yeah, 9th at 7:30. 7:30 to 8:30 is the extra class that I'll be conducting, where I'll teach you guys about the immune system a little bit. And next week the class will be as no, as usual on Tuesday. That'll be the last session for this semester. Okay. All right. Okay. So thanks, guys, everyone, for attending today. Hope to see you in the extra class and in the following lecture, as well, the normal one on Tuesday of next week. All right. Yeah. Yeah. Thank you, guys, for attending and answering questions so nicely. All right. Let me know if anyone has any other questions, by the way. I'm just hanging around to see if anyone has anything that they're confused about. Think everyone's fine then. All right. Okay. See you next class. Bye.